Welcome. We're going to be doing Chapter 17, which is Adaptive Immunity, Specific Defense of the Host. So we're going to be looking at how the host responds from that other wing. Remember, the master has failed more times than the beginner has even tried. I do not remember who this guy is I didn't put his name on, so I apologize. I will have to hopefully find a new... I didn't try to put like famous microbiologists and scientists, and I forget who this is. So this is what we'll be talking about. Let's just remind ourselves the immune system is like a bird with two wings. You got the innate wing and you got the adaptive wing. The innate wing sets the stage for the adaptive wing. So we're going to talk about how this can happen, okay? Now, the adaptive immunity, this relies on T cells and B cells. So this is resistant specific pathogen. So when I say specific pathogen, I can mean this particular strain of this particular virus. That particular specific pathogen. Okay. There is sensitization, and that's when we expose to a pathogen followed by production of antibodies, and there's vaccination. That's deliberately exposing people to harmless versions of pathogens. This does render immunity. Smallpox was the very first disease with the vaccination. What we did is with with smallpox was we got cowpox. Milkmaidens would get cowpox, and they were immune to smallpox. So we gave people cowpox, cow, vaca, vaccination, where we get vaccination from. Alright, so immunology goes back to Louis Pasteur, where he observed immunity in chickens injected with a weakened pathogen. Von Behring received a Nobel Prize for discovery of an antitoxin in 1901. Paul Ehrlich he identified antibodies in serum. Antibodies were made by the body to attack antigens. Antigens are foreign substances that stimulate antibodies. I always say an antigen is anything that elicits immune response. So they are antibody generators. That's where we get the name antigen, is antibody generators. Okay. Now, so basically what happens in our a stem cell can go onto bone and become a B cell, go onto the thymus, become a T cell, migrate out to lymphoid organs or tissues, and fight the good fight. Okay. So anyway, there's specific immunity that is going to rely on the Navy and the Army. Okay. This immunity, we have the Navy, that's our humoral immunity. That's brought about by antibodies. It travels through the body fluids by B cells. B cells are like the Navy of the military. They fight in body fluids or humor. We call that humoral immunity by antibodies. B cells that become plasma cells make antibodies. Antibody-mediated immunity, we call it. Cellular-mediated immunity or cell-mediated immunity is brought about by cell-cell interactions. This is going to be a T cell. Like a T cell recognizes that this is an abnormal cell and kills it. There's two types that are needed to make two types of cells we need. We need an antigen-presenting cell to recognize the antigen, and an antibody-producing cell and a plasma cell. Okay. To my knowledge, every antibody, MADGE, they all come from plasma cells. Okay. Somehow. Antigens, these are going to be produced by their specific proteins found on, like a protein or a polysaccharide that's found on a pathogen. Their substances cause the body to produce an antibody, or they'll produce sensitized T cells, like a memory T cell will immediately become activated, a memory B cells immediately become activated. Antigens are components of microbes. They're found on the capsule, the cell walls. They can be part of the flagella. They can be on fimbriae. They can be seen as toxins. They can be coats of viruses or surface structures. So there are a variety of things that can be seen as antigenic. There are non-microbial antigens like pollen and egg whites, things found on your red blood cells, the A, the B blood type, Rh blood type. Serum proteins from somebody else or another species, that can happen. You've heard of alpha-gal syndrome, where people become allergic to meat. Transplanted tissues or organs, what happens in an organ transplant. Now, what antigens are, we have what's called an epitope, or an antigenic determinant. It's a specific region on an antigen where the antibodies bind. So it's not like the whole antigen, there's a very specific part, okay? I have a way of teaching that to kind of give you guys an idea. A hapten is called a low molecular weight compound that must combine with a carrier to be antigenic. So it's called a hapten, okay? It's got to be on a carrier to be antigenic. And there we have on the right side a hapten. There we have an antigen. So the epitopes, we have an antigen, which is a cell wall component, and then there's multiple epitopes, a triangle, a square, a half circle, okay? Those are all epitopes, okay? Now, there is antibodies. Antibodies are immunoglobulin proteins. Globular proteins that are relatively soluble in the blood. They respond to a specific antigen, and it can recognize that antigen and bind to it. There is antigen binding sites, 2 identical sites that each bind to epitopes. They might bind to the same epitope or different epitopes. Sorry, I need a little drink. I've been recording all morning, and I need my drink. So they are very specific. 2 identical sites that bind to epitopes are your antigen binding sites, so they're made to the same one for these guys here. Okay. Now valence, the number of the antigen binding sites are always the same thing. They have some disulfide bridges here being shown in this antibody. A valence is the number of antigen binding sites that are actually on an antibody. Most antibodies only have 2, so they're called bivalent. But there are some antibodies like IgMs and the dimer IgA. So immunoglobulins, Igs, are constant segments of heavy chains determine the class. There's the order of presentation is MAGE, M-A-D-G-E. IgM is the main attacker. They are the first antibodies that attacks. They're found in blood. They are a pentamer, a 5-antibody collection. IgAs attack pathogens before they enter. They're in body fluids like tears and saliva. They are dimers. They attack before you enter. And IgD is on the surface of a B cell used in B cell sensitization. So I tell it, it's dang it, B cell, get busy. IgG is go get them. They're released from plasma cells to fight microbes. They're the most diverse. They attack bacteria and viruses, so go get them. IgE bind to mast cells to cause allergies or allergic response, so eat you. Okay? So IgG, IgM, IgA, IgD, IgD, MAGE. The IgM is a pentamer. Most are monomers. IgA is a dimer. Okay? All right. Now I'm not going to go into J-tains and secretory component and things like that. All right. Now to remember a lot of the types of T cells, they help me catch some. Helper. Memory is one we don't have here. Help me catch. Cytotoxic. Some suppressor. Then we have natural killer cells, and then we have our B cells. B cells are not a type of T cell, but they become so. Helper T cells activate B cells to produce antibodies. They're going to have a CD4 marker on them. Suppressor T cells combat autoimmunity. They prevent autoimmunity. B cells become plasma cells and activated. They produce antibodies. Natural killer cells or NK cells do immunological surveillance. They police. They destroy abnormal cells. And cytotoxic cells, they destroy abnormal markers. They have a CD8 marker on them. Okay. They're going to release perforin that makes holes in the cell. They're going to have granulozymes that simulate apoptosis. They're going to cause apoptosis, which is programmed cell death, which when programmed cell death happens, the cell that we're going to kill gets bulges called blebbing. And this blebbing prevents the virus from spreading. And blebbing is something that we'll see during a cell going through apoptosis. Now, antigen-presenting cells, what I want to talk about with antigen-presenting cells and antigen presentation is that these are non-lymphocytic. They're going to be a macrophage of some kind or a dendritic. Other cells, they present the antigen fragments after phagocytosis. They display it after being, like if a virally infected cell or bacteria comes in, it displays this abnormal peptide that the infected host cell had. B cells can also present antigens on an MHC a little bit. They're weird. We'll talk about them doing that. These are found in organs, tissues, fluids, and what's called an M cell. An M cell is a microfold cell, or an M cell. They allow transfer of pathogens to lymphocytes and antigen-presenting cells in your gut. Peyer's patches have them. Lymphoid tissue in the small intestine that protects bacteria from the large intestine bacteria. They have a high degree of M cells. Okay. Now, there are class 1, class 2 MHCs. Okay, I want you guys to know these. Class 1 MHC is made by all nucleated cells. They bond to any cell, a T helper cell that has, or cytotoxic T cell, I apologize, that has a CD8 marker on it. MHC1 goes with a CD8 marker. Antibodies are not produced, the infected cell is destroyed. MHC2 tells us, hey, this antigen is dangerous. Now, MHC1 says, hey, I'm an abnormal cell, I'm infected, kill me. MHC2 says, hey, this antigen is dangerous, get rid of it, any of them like it. It's only made by immune cells. They have CD4 markers on their T cells that respond to it, and antibodies are produced. Okay, use the =8 rule. CD8 times MHC1 equals 8. CD4 times MHC2 equals 8. Okay, remember the =8 rule. I will ask you, what does an MHC bind to? CD8. What does an MHC2 bind to? CD4. Okay, I will ask that on a test. B cells in humoral immunity, there's T-dependent antigens. They are antigens that are actually presented with MHC to a T helper cell. T-dependent antigens. A T helper cell produces cytokines and it activates the B cell. Then there's T-independent antigens. They stimulate B cells to make antibodies with no T cell required. Okay. What we can see is I have a T-independent antigen can show up, activate my B cell and sensitize it, and then a helper T cell can turn it on. Okay. Now, B cells differentiate into plasma cells, antibody producing, and memory cells. The plasma cells are active, the memory cells are inactive. The plasma cells make antibodies, the memory cells just remember, and as soon as they see this antigen, they get activated. Now, what I'm going to talk about is B cell activation. Now, what I'm going to do, I kind of like how our book does it versus A and B. Now, so let's do B cell activation. Okay. Here we have a B cell. Okay, this is a B cell. Now, B cells have on them an antibody called an IgD. And this IgD is going to act as a receptor. Now, let's say we have a bacteria here, and this bacteria has various epitopes. One's a square, one's a circle, one's an X, and one's a triangle. Various antigens, okay? These are antigen epitopes, okay? Epitopes of antigens, okay? Different antigens, different epitopes, okay? Now, what we're going to do, that shows up. What this B cell is going to do is take this antigen and internalize it. Then what it's going to do is it's going to make a molecule called an MHC2. Remember, that's released from immune cells. and it's going to have this antigen on it. Okay. Now what we're going to basically do with this guy is it's now sensitized. Okay. Now that we've sensitized it, we can then activate it with a helper T cell. Okay. And we're going to see when that happens in just a little bit. Okay. Sorry, I realized I didn't have that on the right screen. That's what I drew out. Sorry, I just realized I did not have that on the screen. The captures. I have two screens, and anyway, go ahead and pause that so you can draw it. That's how we do it, okay? Alright, so basically what happens here is we have a pathogen that can show up. It can be floating around. That's one of the ways it can show up. It binds to the IgD. The MHC will then show up and display the antigen. Then what's going to happen is next, and I didn't think my notes went into that yet, is a helper T cell will come in and activate it, and it will make antigens. Okay, so I did talk about it. I couldn't remember my notes was covering that because I was looking at my screen. So I'm going to show you how the T helper cell gets activated next. Okay. Now, clonal selection is the development of clones of B cells and T cells. Now that we've made this B cell, we can clone them several times and make a bunch of them and really make a bunch of antibodies. We can clone these. Now, antigen-antibody binding. We made the antibody. We bind it. Antigen-antibody complex, where that binding occurs. It has affinity. There's a strength of bond that is there. Its specificity. It recognizes the shape of the antigen's epitope. So, for example, I had a square antigen. I had an X. I had a circle. And I had a triangle. It only recognizes square. Okay, it's that specificity. When it binds to it, and the foreign organisms or toxins can be rendered harmless by being agglutinated, clumped together. Opsonization, where it's going to be coated and enhancing phagocytosis. It's going to have complement stick to it, which is going to happen as well. Opsonization, cell-mediated cytotoxicity. We're going to kill the cell, or we're going to neutralize it. We're just going to coat it, and the cell can't adhere, so it can't make you sick. Okay. Remember, we have to have those receptors bind to like those PAMPs and stuff involved in making you sick. And those binders and those receptors and the fimbriae and stuff, bacteria have to stick to you, make you sick. So agglutination is when antibodies cause them to clump together, makes it easier for bacteria to eat it. Opsonization is coating the bacteria with antibodies, making it easier to be phagocytosed. Okay, antigen is coated with antibodies and complement, and it enhances ingestion and lysis, makes it easier to phagocytize. Complement will bind to it, and when complement binds to it, it activates it, and it can form an MHC and create pores. Now, antibodies are made, so antibodies are also helping the innate immunity. So these two immunities help each other. That's why I say it's a bird with two wings. Without both wings, it can't fly. Antibody-dependent cell-mediated immunity. The antibodies are going to bind. It's going to destroy. It's going to cause cells to release perforin and other lytic enzymes and destroy a target cell, like a parasite or a antibody can bind to it and help damage, cause complement, cause immune cells to come in, cause a variety of things. So anyway, all right, neutralization. Antibodies can inactivate microbes to block attachment, or they can neutralize toxins. They can bind to toxins. Now the toxin can't, the AB toxin, things like that, can't do anything. Now let's talk about CD4 T cell activation. Now CD4 T cell activation is going to be the one that we use, the helper. Then we're going to do cytotoxic T cell activation. So let's talk about CD4 T cell activation, and let's get into that, is how we turn on CD4 T cells. Okay, how is the CD4 T cell activated? Alright, so what we're going to do, we're going to have a macrophage, okay? And this is going to be our macrophage, which is an antigen presenting cell, or APC, okay? Now I'm going to go through the whole process. Let's say we have that same bacteria from earlier, okay? Same bacteria from earlier, okay? It has square antigen, it has circle antigen, X, and triangle antigens on it, okay? Now this bacteria, this antigen presenting cell, will engulf it, will phagocytize it, okay? Phagocytosis happens, and it's brought into a phagosome, okay? We have our phagosome. Now our phagosome here, okay, what we're going to do is we're going to bring this in and we're going to gobble up this bacteria, okay? And we're going to leave behind these antigens. Okay, we're going to leave behind these antigens. Then I'm going to give you an analogy for this, okay? Now, what we're going to do, we're going to take one of these antigens. Now, I want to grab square antigen and grab it. We produce this thing called an MHC2. But we grab square antigen. Okay, now remember my B cell is activated to square antigen. Okay, now we have some T cells over here. CD4 T cell. Okay, CD4 T cell. So this is a CD4 T cell. Okay, and let's get about 3 or 4 of these. Let's get about 4 of these. Okay, CD4 T cell. These are all CD4 T cells. Okay. Now what we're going to do is they need a T cell receptor. Okay. So this one here is triangle. This one here is X. This one here is square. And this one here is circle. Okay. These are called T cell receptors. Okay, TCRs, T cell receptors. Now, what we're going to do is this MAPC is going to come in, and it's going to take this antigen that is displayed and take it and be like, nope, I don't fit there. It's going to take it here and be like, nope, don't fit here. It's going to take it here and it's going to be like, oh, I do fit. And it's going to activate this T cell into a helper T cell. Now this helper T cell that it becomes, it's now activated to that square antigen, that epitope. So what's going to happen is, it's now going to go find that B cell from earlier and activate it to become plasma cells and be able to make antibodies that way. Okay? That's what's going to happen. Now I want you to think of it like this. Think about this bacteria here is Cinderella. And Cinderella is going to the ball to dance with Prince Charming, or a Prince Charming, and find her love. She goes to the ball. The clock strikes midnight. Her gown turns back to rags. Her tiara turns back to whatever it was. Her carriage back to a pumpkin. She drops the glass slipper and runs away from the ball. Okay? Prince Charming picks up the glass slipper and says, I want to find the girl that fits this. So he goes out in the town to find the girl that fits, and then he finds her. And he's like, that's his girl, that's his Cinderella. They're going to sweet talk, they're going to love each other. And this helper T cell is activated. Okay? This is called co-stimulation. So an antigen is presented on an antigen-presenting cell ingested microorganisms. Antigen gets processed into various antigens. It's combined with the MHC2 displayed on the surface. A T cell receptor and a CD4 T cell binds to the MHC and antigen complex. This has a toll-like receptor and things like that. So it secretes co-stimulation molecules, activates the helper T cell to make cytokines. The cytokines will help to co-stimulate. Cytokines help the helper T cell proliferate and develop into a helper. Then the helper goes over here and finds the B cell, activates that B cell to the antigen, and makes antibodies. Okay? Think of these two processes happening in parallel to each other. Okay? All right. Now, so that's how that happens, and that's what I drew out. Okay? All right. Now, cytotoxic T cell activation. Normal cells aren't going to do this, but a virally infected cell or cancer cell will. So let's talk about this a little bit. Okay. Now, I'm going to draw that. So let's say I have a little beautiful little cell here. Okay, like a kidney cell. All right. Here's the nucleus. Okay. Now we're going to do this like it's a virus. Okay, we're going to get a virus here. So we have a virus. I'm going to talk a little bit more than what the notes have. This virus has some kind of nucleic acid in it. Has some kind of nucleic acid going on in there, okay? DNA or RNA. Okay. Now what's going to happen is we're going to come in. This DNA is going to shoot into here, and it's going to come in and hijack this cell. Next thing you know, this cell is producing virus bits. Okay? Viral proteins. It's making viral proteins. This cell is going to make an MHC called an MHC1. And this MHC1 is being displayed, and this is like waving a red flag at a bull. Then what we have over here is a CD8 T cell. This CD8 T cell has a T cell receptor, a TCR, okay, and that CD8 marker, okay, is on it, and it will bind, and what will happen is it will be activated and become cytotoxic. And what that will do is lyse the cell. It will cause lysis, and we will destroy this virally infected cell so the cell doesn't spread the virus. Okay? And pop it. It's going to pop it like a balloon. Okay? Now, what's going to happen? The normal cell doesn't trigger this, but a virally infected cell will. They will produce abnormal endogenous proteins called antigens, which those right here, by the way, these are your endogenous proteins. Okay? Those are endogenous proteins. Okay? Alright, so what's going to happen is the abnormal antigen gets presented on MHC1, a CD8 T cell with a T cell receptor that matches that antigen, bind to it. It becomes cytotoxic, and the cytotoxic T cell will destroy the infected cell by apoptosis, popping it like a balloon. Okay, so here we have an antigen presenting cell. Okay, well, we don't have that there. Sorry, I forgot. This is the a virally infected cell. I said antigen presenting cell. I apologize. We have a virally infected cell, and it takes the antigen, presents it, and then a cytotoxic T cell is produced. It then will go and lyse and kill the infected target cell. The needs of the many outweigh the needs of the few. Now, one of the things we can do with cancer cells is, tumor cells, cancer cells, is we take our lovely little natural killer. It's out there policing the tissues. doing surveillance. And it's like, wait a minute, this is an abnormal cell. You have abnormal proteins on you. You ain't right. So it recognizes it. It adheres to it. Recognizing and adhering. Then it will take its Golgi, which was here, and aim it. Then it will release perforin and put a bunch of holes. If you put enough holes in something, you'll kill it. I'm going to start using that when I say immunological surveillance. I'm going to say we're policing tissues in AP2. Okay, it does not need to be stimulated by an antigen. Okay, it's non-specific. Perforin doesn't need, so it can be an abnormal cell, but natural killers. Chemical messengers of the immune system are cytokines. They control communication between interactions called cytokines. There's a cytokine storm. This is when the cytokines produce more cytokines, harmful, damaged tissues. That's what killed people during COVID. There's cytokines. There's interleukins. They're between leukocytes. There's chemokines. There'll be a cytokine that induces the migration of leukocytes in the area. My chemokines are the macrophages of the area, and they're like, I'm going to phagocytize, and you dang right, they're going to phagocytize. Okay, chemokines, they induce migration of leukocytes, especially macrophagic leukocytes. Interferon is a chemokine, a cytokine that are produced by virally infected cells. Tumor necrosis factor induce inflammation and autoimmune diseases. Hematopoietic cytokines control pathways of stem cell development, like red blood cells and white blood cells. Cytokine storm is when you produce so many. That's what killed people during COVID, and it's what killed people during the Spanish flu. Antibody titer is the amount of antibodies in serum. How many antibodies do you have? Your antibody titer goes up during your immune response. The primary response after initial contact with the antigen, it's very slow. Antibiotic production is slow. You don't have antibodies for 7 to 10 days. It's slow to rise in titer, and it peaks after 10 to 17 days. Secondary response, because there's memory here, second exposure, memory cells will immediately produce antibodies, and that'll peak 2 to 7 days at a greater magnitude. So the secondary response always gives us a greater amount of response. You produce IgMs during the secondary response earlier. You produce IgGs earlier and greater during the secondary response. Both make IgMs for about the same level, but you make IgMs earlier during the secondary response. Okay? And here you can see it there. Okay? Alright, now, I do like to ask the difference in primary response and secondary response on the test. This will be on the test, naturally acquired versus artificial acquired immunity. Natural acquired immunity, you get it naturally, as the name says. This develops as a person exposed to antigens gets ill and then they recover. That's naturally acquired active immunity. I get sick, I fight it off, I get it. That's naturally, okay? Naturally acquired passive immunity, I would get like through my mother's milk or a placenta. Artificial immunity, there's artificial acquired. That's from a vaccination. That's not natural. Vaccinations are things we develop. Antigens are in the blood. Artificial acquired passive immunity is injection of antibodies. Then we use antibodies. We can get it from an animal like horses. We take a horse, we shoot the disease into the horse makes antibodies. We take the antibodies, we remove them from, and we give them like the monoclonal antibodies that we use to treat COVID. Okay? So I might say which of these is an example of artificially acquired passive immunity. Or give an injection of antibodies as an example of A, B, C, D. Okay? Alright, that's it, and I'll see you in the next one.